Hello, my name is Dr. Corey Rokas, and I am a veterinary clinical pathologist and a member of the digital cytology team with Zoetis. I'm on the other side of your Vetscan Imagyst, reading your hematology and cytology samples. Today's videos are going to be some tips and tricks just to help you improve the quality of your hematology and cytology samples so we can help you do the best for your patients. Today, what we're going to start with is some blood smear tips, preparation, including submission. So let's go ahead and get started. First, what should an ideal blood smear look like? This was always the challenge for me, figuring out what my blood smears had to look like. So what we want to aim for is this really nice candle flame shape we see here. We have our drop of blood near the label of the slide, and then going forward, we go into the body, and then the smear will taper up into a monolayer and a feathered edge. And the feathered edge and monolayer are where you're going to be doing the bulk of the work in the blood smear. So that's what we want to make sure that those parts are there and visible. So how do we get that shape? Let's take a peek at an ideal blood smear preparation. To start with, you want to make sure that you label your slide, patient name, date, so that we know which patient this blood smear came from. You want to take a small drop of blood and put it near to the label, not touching, but maybe about a few centimeters in front, 2 to 3 centimeters. And I like to do a capillary tube. So I would take a capillary tube, dip it into my EDTA sample, tap out a really small drop of blood, about 2 to 3 millimeters, no larger than the size of a pencil eraser. What you're going to do next is, which was always the challenge for me, is take your spreader slide, drag it back slowly until it touches the drop of blood, so where it spreads out all the way along the length of your spreader slide. Once that blood is all spread out, go ahead and spread forward in a really nice, quick, smooth motion. And to sometimes get a better feathered edge, you can do a little lift off at the end of the smear, and that can help create that really nice feathered edge. If you're having trouble with that, or if you're pushing down really hard, like I used to do, I would prop this slide with the blood up on my finger a little bit so that it gave it a little bit of an incline, and then I would smear up. So that way I wasn't pushing down too hard, which was really helpful for me. In terms of the spreader slide, you want to hold it ideally at about a 30 to 35 degree angle. If your patient is anemic, however, like those IMHA patients with hematocrits of 15%, you want to go ahead and increase that angle to about 40 to 45 degrees. That's going to make our smear a little bit thicker and hopefully a little bit more readable. So we want to make sure that we're making the most out of a small watery sample. This does take practice. So this is something that you want as a few members of your team to be able to do, and you want to just keep doing them regularly. Even if you're not looking at them to start with, make sure you're going through the procedure so that you can make sure you're getting that really nice candle flame shape. Let's go through a few mishaps. Let's see what happened. So with this, what happened with this smear is we just had too much blood. And again, you don't want your drop of blood to be any larger than a pencil eraser, because otherwise it's just going to be too thick and it's going to be too messy and it's just not going to smear well. Next, let's take a look at this sample. This one is, again, look at that size of that blood drop. That's huge, probably about 2 or 3 pencil erasers. So we had a little bit too much blood to start with, pushed forward, and then instead of lifting off at the end to get that really nice candle flame shape, I can tell that the slide kind of stopped, because can you see that thick, really thick line at the far end of the smear? So we don't have a feathered edge here. And again, this comes back to being just a little bit too much blood for this sample. Where are we going to scan? I think once you have your blood smear made, this is going to be the most important part, because you want to get the most information out of the blood smear that you can. So ideally, what you want to do is scan the entire slide, either if it's with your eyeballs in your practice before you send it to me, or with the Imagyst scanner. Ideally, we want to scan the entire blood smear. We can get information from the feathered edge, from the monolayer, and from the body. But sometimes time is of the essence, right? You don't have time to sit there and scan through the entire smear. Totally get it. So at the bare minimum, what you want to make sure that you're scanning is the entire feathered edge and the entire monolayer, and not just a strip of each, because we can miss information. And in the image, where that feathered edge, it's that second image that says scan 1 under where do I scan, and that's showing you where your feathered edge and your monolayer are. Typically, that is about the first quarter to third, depending on how long your blood smear is. of your blood smear. The feathered edge is where we're going to look for platelet clumps. We're going to look for blood parasites out there. We can look for neoplastic cells because they're all heavy, so they get pushed to the feathered edge. And in the monolayer is where you're going to be doing your differential. You're going to be looking for red cell morphology, white cell morphology. Platelet estimates are in the monolayer. So at the very least, you want to make sure you've captured the entire feathered edge and the entire monolayer for review. For any blood smear that you're submitting, to make those blood smear reviews pack the most punch, you want to always make sure that you are including your CBC data along with it. A blood smear without a CBC is kind of like driving a car with no lights on. At night, you kind of have an idea of where you're going, but you don't have the full picture. So with the CBC, we can help identify discrepancies between the analyzer data and the blood smear itself, and vice versa. We can see things on the blood smear that aren't on the CBC, like nucleated red blood cells, neoplastic cells, parasites, things of that nature. So the CBC and the blood smear really are the best of friends, and we don't want to break them up. So make sure that you are submitting blood smear CBC data with every blood smear that you are sending in. A few pitfalls that happen with blood smear submission. What you want to do with your blood smears is make sure that you're making a smear right away. So the minute you're drawing the blood out of that patient, go ahead and make your blood smear. You actually don't have to stain it right away, but you want to get that smear made, because a lot of times there are some species of blood parasites that once they get into the EDTA tube will actually fall off. The big one is mycoplasma, even with a matter of minutes. So especially if you have an anemic cat that you're worried has mycoplasma, you want to make a blood smear right away. If you're sending that blood out, by the time it gets to a lab, the little critters are going to be in the tube and we're not going to be able to see them. So make sure that you're making your blood smear absolutely right away. In terms of drying, we mentioned before, you want to make sure that your blood smear dries completely before you stain it. And why we want to do that is to avoid water artifact, which you can see in the smaller image here. That creates kind of little hole punches. The water gets trapped against the red cells and it makes little hole punches. And under the microscope and on the Imagyst, they can actually mimic blood parasites, kind of terrifying a little bit to try and parse out whether it's an artifact or not. And the only way you're going to get this water artifact is if you are staining your smear before it's completely dry. So make sure you're letting it dry. Room temperature sitting on the tabletop is absolutely fine. If you're pressed for time, a hair dryer set on the cool setting can speed up that process. But we also don't want to dry it too fast, or then we can make the red cells all shrink up. When you're collecting your sample, you want to make sure that you are filling your blood tube right to the fill line on the EDTA tube. If that is underfilled, you're actually going to create red cells that get shrunk and spiky balls and turn into echinocytes. And that's a hard artifact to read through as well sometimes. Excuse me. The larger image that we see here is actually stain precipitate. And when your stain is overused and isn't freshened regularly, we can get stain precipitate that creates a film over on the top of the stain. That can also mimic bacteria. So we don't want that. So you want to make sure that you are changing out your stain at least once weekly, or depending on how the manufacturer recommends. If you are a frequent flyer, you want to make sure that your cleaning out your stain more frequently, and you want to have a clean stain for hematology and cytology samples, and a dirty stain for things that are going to have bacteria. And I always remembered it as ears and rears. So when you have fecal samples, if you're looking at those, or you're looking at ear cytologies, that's what you're going to want a separate set of stain for. So I hope you were able to take a few tips from this video to help improve your blood smear technique, learn where you're going to scan and what's important for us for the images, and how to avoid some common artifacts that we see on blood smears. So thank you so much for listening, and we'll see you next time.